Welcome back, Focus Learners. Today, we're jumping into a truly fascinating explainer, covering the biological architecture of one of the world's most intensely cultivated plants. We are going all the way from the absolute baseline of plant genetics, right up to the cutting-edge, laboratory-level science of seedless cannabis. Honestly, whether you're a commercial cultivator or just someone who geeks out over agricultural science, this explainer is going to completely change how you look at crop development. Okay, let's dive into this. Here's our roadmap for today. We'll start with the genetics of cannabis, move on to creating a stable strain, explore the magic of feminization, and finally wrap up with triploids and seedless cannabis. Let's get started. So, Section 1, the genetics of cannabis. To really grasp the engineered future of cultivation, we have to start at the very beginning. We need to separate what a plant is on a cellular level from what it actually looks like in the real world. Think of the genotype as the invisible genetic blueprints. The phenotype, on the other hand, is what you observe with your own two eyes. The size, the bud color, the leaf shape, all of that. But here's the kicker. That exact same genotype can express completely different phenotypes depending on its environment. Things like light, temperature, and nutrients play a massive role. You could literally have the best genetics on the planet, but if your environment is unstable, your physical expression is going to be unstable too. So the crucial point is how we actually achieve genetic stability. Plants have these alternative versions of genes called alleles. When a plant is heterozygous, it's holding two different alleles for a specific gene. If you breed that plant, the offspring are a complete roll of the dice. It's totally unpredictable. The absolute ultimate goal for a breeder is a homozygous plant. That means it has two identical alleles for a gene. If it has two alleles coding for, say, purple flowers, it's going to reliably produce purple flowers. Breeders are constantly hunting for these homozygous plants to make sure their strains are totally uniform and ready for the market. And all of this complex genetic information, it's packed into a very specific, tiny space. Cannabis plants are naturally diploid organisms, meaning they possess exactly 20 chromosomes. So when standard reproduction happens, the pollen from the male and the ovule from the female each contribute exactly half, 10 chromosomes from each parent, to form the brand new genetic identity of that offspring. All right, moving right along to Section 2, creating a stable strain. Think about it for a second. Let's say you have an award-winning, incredibly high-potency plant. If you just cross it with another random plant, you're going to get this unpredictable polyhybrid soup of genetics. Your chances of finding that exact same incredible plant in the offspring seeds? Pretty much slim to none. So how exactly do breeders bridge that huge gap from wild, unpredictable polyhybrids to the incredibly uniform strains we see on commercial shelves? While breeders basically rely on three main strategies. Now sure, some simple traits like leaf shape follow basic Mendelian inheritance. They're just controlled by a single gene. But the traits commercial growers are really chasing, things like yield, potency, and terpene profiles, those are polygenic. They need multiple genes working together perfectly. To lock those in, breeders use selfing, which is literally pollinating a plant with itself. This bumps up stability by 50% per generation, but the downside is it causes rapid inbreeding depression, making the plant pretty weak. Then you have backcrossing, where you take the offspring and cross it back to a parent to fix a very specific trait. And finally, the holy grail, true F1 hybrids. This takes two totally different, highly stable inbred lines and crosses them. It takes years of rigorous work, but it produces unbelievably vigorous and uniform crops thanks to a biological phenomenon called heterosis, or hybrid vigor. Which brings us to Section 3, the magic of feminization. If you just go out and plant regular seeds, basic biology dictates a 50-50 split between male and female plants. Now, for a commercial grower, that is a massive, highly stressful liability. Male plants produce pollen, not the THC and CBD-rich flowers that everyone actually wants to buy. If even one rogue male manages to pollinate your crop, the quality of your flower just plummets. You basically have to maintain constant, paranoid vigilance to spot and destroy those males before they mature. Feminized seeds, though, they completely change the game. They give you a 99% guarantee of producing female flowers. When you're an operator managing thousands of plants, that kind of genetic insurance is absolutely crucial for a bountiful harvest. But it begs the question, how do you actually force a seed to be female? The secret is a chemical compound called silver thiosulfate, or STS. You see, the plant relies on a hormone called ethylene to signal the development of female flowers. STS acts as a straight-up chemical blocker. You spray it on the plant, it binds to the ethylene, and completely suppresses it. And this triggers a deep evolutionary survival mechanism. The plant basically thinks, oh no, I can't reproduce normally. So the biological female desperately switches gears and starts producing male pollen sacs just to keep its genetic line alive. It's a total biological hack. Let's move to and see how this builds in practice. Breeders will take a known female plant, which, remember, biologically only has 2 X chromosomes, and spray it with STS during its dark cycle so the lights don't degrade the chemical. The ethylene gets suppressed, and boom, male flowers appear. But here is the absolutely brilliant part. Because this specific plant is biologically female, it physically does not even have a Y chromosome to give. So the pollen it drops, it only contains X chromosomes. When you take that exact pollen and use it to fertilize another female plant, the resulting seeds literally have no choice but to be female. And finally, we arrive at section 4, triploids and seedless cannabis. As we talked about earlier, standard plants are diploids, or 2N, meaning they carry 2 sets of chromosomes. But by using some pretty advanced breeding techniques, cultivators can artificially induce chromosome doubling. This turns the plant into a tetraploid, or a 4N plant, with 4 complete sets of chromosomes. Now, why go through all that trouble? Because changing those chromosome sets has an immediate, massive impact on a cellular level. Tetraploids have physically larger cells, which means thicker stems and bigger leaves. But the real magic is the increased gene dosage. If the genes responsible for making cannabinoids, like THCA synthase, exist in 2 copies in a normal plant, well, a tetraploid might have 4 copies. You are literally doubling the biological machinery available to pump out those active compounds. But honestly, tetraploids are really just a stepping stone. The ultimate commercial goal is the triploid. To get there, you take that artificially created tetraploid with its 4 sets of chromosomes and you cross it with a regular old diploid that has 2 sets. The offspring from that pairing inherits an odd number of chromosome sets, making it a triploid or 3N. And look, if this sounds like wild science fiction, it's actually incredibly common in everyday agriculture. This is the exact same biological architecture used to grow the seedless watermelons and seedless bananas that are probably sitting in your kitchen right now. And this brilliantly illustrates why this is such a big deal for commercial growers. John Keating, a top genetics expert from Tesoro Genetics, who's really pioneered triploid cannabis, sums it up perfectly. He notes, when you remove reproductive pressure, the plant can redirect more energy toward resin production and secondary metabolites. By creating a triploid, you are rendering that plant biologically sterile. It physically cannot produce viable seeds anymore. So instead of wasting all that precious biological energy trying to reproduce, the plant hyperfocuses all of its energy into pumping out resin, terpenes, and cannabinoids. The commercial benefits of this are, frankly, revolutionary. You've got a sterile crop, which instantly, completely wipes out the devastating risk of a rogue male plant ruining an entire facility's harvest. You've got that increased gene dosage we talked about, pushing potency to the absolute maximum. You've got larger cellular structures, creating just massive flowers. And the real-world data backing this up is staggering. We're talking about large-scale commercial operators, not just tiny isolated labs, reporting dramatic yield increases. We are seeing production levels approaching an unbelievable 6 pounds per light. That is huge. However, we do have to be real about the risks with this science. Polyploid breeding is incredibly difficult and highly unstable if it isn't managed perfectly. When you force a natural diploid into a tetraploid state, you are actively pushing it against its very nature. If every single cell doesn't successfully convert, you end up with what's called a mixoploid. It becomes this unstable biological mosaic of 2N and 4N cells. Because the plant naturally wants to be 2N, those diploid cells will eventually try to dominate, and the plant will literally try to revert back. Imagine the sheer panic of watching your meticulously engineered crops start to mutate in real time. This kind of physiological stress can lead to awful germination rates, hermaphroditus, and severe mutant phenotypes. It takes incredibly precise environmental management to keep these enhanced organisms stable. Which brings us to our final thought for this explainer. We've traveled from basic Mendelian inheritance to hacking the plant with hormone blockers, all the way to engineering sterile, hyper-producing triploid superplants. The science of agricultural genetics is moving at absolute breakneck speed. So as we master polyploidy and start stepping into the era of direct gene editing, we have to ask ourselves, what does the future landscape of cultivation actually look like when these heavily engineered, massively high-yielding plants become the undisputed baseline? It's a wild evolution of control, and it's absolutely going to reshape the industry forever. Keep learning, stay focused, and we'll catch you in the next explainer.